In this video, we're going to cover Zwitter, looking at what amino acids are, the structure and reactions of amino acids, how to name them, what Zwitter are themselves, and how to do thin layer chromatography of amino acids. Proteins and enzymes, so looking at what peptides are, the different types of protein structures and the intermolecular forces present in proteins, and then what enzymes are and how they work. And DNA and anti-cancer drugs, so looking at nucleotides and DNA structure, polynucleotides and anti-cancer drugs. We're going to start by looking at what amino acids are. Amino acids contain two functional groups: an amino group, which is an NH2 group, and a carboxylic acid group, which is a COOH group. They can also contain different R groups. We'll look at these in more details throughout this topic, but each amino acid will have a different R group. You can find a list of common amino acids on the back of your chemistry datasheet. This is the structure of an amino acid. You can see the amino group, the carboxylic acid group, and the R group. Now the R group could be lots of different things, and we're going to look at some examples of these throughout this topic. Amino acids are chiral molecules because the centre carbon has 4 different groups attached to it. So a solution of a single amino acid enantiomer will rotate plain polarised light. We can see the 4 different groups quite clearly if we draw the amino acid like this. And we can draw both enantiomers by drawing the amino acid out in a tetrahedral structure, adding in a mirror line, and drawing the mirror image of that amino acid to show both enantiomers. Amino acids are also amphoteric. This means that they have both acid and base properties. They can act as acids because the carboxylic acid group can donate a proton, and they can act as bases because the amino group can accept a proton. So for example, this equation here shows how the carboxylic acid group can donate a proton and how the amino group can accept a proton. Remembering, a proton is just a H+ ion. We're now going to look at how to name amino acids. There are 4 stages to name amino acids. Find the longest chain that includes the carboxylic acid and name it. Number the carbon atoms, starting with the carboxylic acid being carbon number 1. Add in the amino groups, add in any other groups. This is quite a simple amino acid, but this process would work for any given amino acid. You'd start off by identifying what carbon staining you're looking at. So in this case, we've got 2 carbons in the chain, so the base of the name would be based on ethane for 2 carbons. We'd then look at where the amino group is. So our amine group is on carbon 2, because as this is a carboxylic acid, this carbon here will always be carbon number 1, regardless of the length of any of the chains present. So putting these parts of the name together would give us the molecule 2-amino-ethanoic acid. Amino acids also have common names, and you'll see these on the back of the datasheet. So this amino acid is also called glycine, but its chemical name would be 2-amino-ethanoic acid. This amino acid is slightly more complicated, but would name it in exactly the same way. The longest chain, starting from our carboxylic acid carbon, has got 4 carbons in it, so its name will be based on the stem of butane. The amino group is on carbon number 2. Again, remembering the carboxylic acid group will always be carbon number 1. And we also have a methyl group on carbon number 3. So putting these different parts of the name together, our molecule will be called 2-amino, 3-methyl, butanoic acid. A Zwitter ion is a dipolar ion. This means that it has both a positive and a negative charge at different parts of the molecule. Zwitter ions also have isoelectric points. An isoelectric point is where the overall charge on the amino acid is nearly 0. Each amino acid will have a different isoelectric point because each amino acid will have a different R group. Zwitter ions can only exist near the isoelectric point because if the overall charge is nearly 0, therefore you must have both ions present, both the positive and the negative charge present. We're now going to look at some examples of what Zwitter ions are. For reference, I've drawn the general formula for an amino acid. We'll start off by looking at what the Zwitter ion is at the isoelectric point. So this is where it will be a dipolar ion with a positive charge and a negative charge. So this will be formed at the pH where the overall charge is 0. This is the structure of the Zwitter ion at the isoelectric point. So it's the pH where all of the NH2 groups have been protonated to NH3 groups, and all of the carboxylic acid groups have been deprotonated to our carboxylate ion groups. Remember, amino acids are amphoteric, they can act as acids and bases, so this is the pH where it's acting simultaneously as a base, accepting a proton, and an acid, losing a proton. We also need to know what happens if we're at a pH which is more acidic than the isoelectric point. This is the structure of the Zwitter ion if we've got a pH more acidic than the isoelectric point. In this case, because we're more acidic, there are more protons present, we'll make sure that all the NH2 groups have been protonated to NH3 groups, but all of our carboxylic acid groups will remain unchanged. We can also have Zwitter ions at a more basic pH than the isoelectric point. And this is what that Zwitter ion would look like. If we are more basic than the isoelectric point, the NH2 groups will be unchanged, but all of the carboxylic acid groups will have been deprotonated to our carboxylate groups. Because in basic conditions, we will deprotonate the acid group, and there are obviously no free protons to protonate the amino group. Thin layer chromatography, or TLC, can be used to separate and identify different amino acids. You'll have studied thin layer chromatography during your GCSE work, and you'll look at this in more detail in a future topic in A-Level Chemistry, but as a quick reminder, you have got a chromatography plate, a baseline where you'd put your mixture of different components, you'd place this in a solvent, and as the solvent moves up the plate, it separates the different components by their relative solubility in the solvent compared to their affinity for the stationary phase. Like I said, you'll look at this in much more detail in the separate topic dedicated solely to using thin layer chromatography. So we can use this to separate a mixture of different amino acids. As we've already looked at, different amino acids will have different R groups, and those R groups mean the amino acids will have different solubilities in the same solvent. Therefore, they can be quite easily separated by thin layer chromatography because each amino acid will have a different solubility in the same solvent. Therefore, each amino acid will move up a different amount up the thin layer chromatography plate, therefore allowing them to be separated. That's as much detail as you need to know for this topic. A slight problem with doing this process practically, however, is that amino acids aren't coloured chemicals, so they won't show up on your thin layer chromatography plate. So there are a few different methods you need to be aware of for how you can visualise, how you can see the different amino acids on the plate. The first way you can do this is by spraying a chemical called ninhydrin solution on the TLC plate. And this will turn the amino acids purple so you can see them. You could also use a TLC plate which has been coated with a fluorescent dye. If you place this TLC plate under some ultraviolet light, the spots will glow, and then you can draw around them so you know where the spots are. Or you could put the TLC plate in some iodine vapour. Like with an ninhydrin spray, this will stain the spots purple. We're now going to look at what peptides are. A protein is a condensation polymer of amino acids. They are made up of lots of amino acids joined together by peptide links. So here we've got two identical amino acids just written in their general formula, and we can join them together through the condensation polymerization process. Condensation polymerization is where two molecules join together through the loss of a water molecule, hence the name condensation. This is where the two amino acids will join together. We're going to join the carbon on one of the amino acids from the carboxylic acid group with the nitrogen on the second amino acid. We can see here that by losing the OH from the carboxylic acid group and the H from the nitrogen, that is where our water molecule will come from. So if we join these two molecules together, we will have our dipeptide molecule. This is the peptide link. This is where our new bond has been formed. Again, we can see how we've joined the carbon from the carboxylic acid group with the nitrogen from the amino group. We formed a molecule of water in this process, so reacting these two molecules together is a condensation process. If we wanted to split these two molecules apart, break them back into two individual monomers, that would be a hydrolysis process, the reverse of the condensation process. We would need to add in water to make that reaction happen. If two different amino acids are joined together, there are two possible dipeptide products. Remember, because of the R groups, you can have lots of different amino acids. So our first amino acid has got a CH3 group as its R group. And our second amino acid has got a hydrogen as its R group. We could join both of these molecules together to form this dipeptide. We've essentially joined the molecule below to the right hand side of our first molecule, so we could join them together like this. Or we could join them together the other way around, so the R groups have been reversed. Now these are two different molecules. To satisfy yourself of this, on the first molecule, the R group where the CH3 group is, so the CH3 group is closest to the amino group at the end of the molecule. In our second example, this CH3 group is now closest to the carboxylic acid at the end of the molecule. So these are two distinct products. Be careful with this, because an exam question might ask you to draw both products which are formed when these two amino acids join together. Now, the question is referring to drawing both different dipeptide structures. When it says draw the two products which are formed, it doesn't mean, for example, draw this molecule and maybe a molecule of water or something like that. It means draw the two distinct dipeptides you would form from the same two amino acids. We're now going to look at what proteins are. Proteins have three different levels of structures. They have the primary structure, the secondary structure, and the tertiary structure. The primary structure is the sequence of amino acids in the long chain that makes up the protein, the polypeptide chain. It's essentially just one long sequence of amino acids. Peptide chains can form hydrogen bonds with each other. The chain isn't just a straight line. The shape of the chain is called its secondary structure. The most common secondary structures are an alpha helix, so a bit like a coiled spring, and a beta pleated sheet, a bit like a piece of paper folded up. These are the kind of structures which are commonly referred to as secondary structures. The amino acid chain itself is then often coiled and folded in a characteristic way. Extra bonds can form between different parts of the polypeptide chain, and that will give the protein a unique 3D shape, which is its tertiary structure. So for example, this diagram shows an alpha helix chain coiled around itself into a certain tertiary structure. The secondary and tertiary structures are formed by intermolecular forces which cause the chain to twist and fold. These are primarily hydrogen bonds and disulfide bonds. Temperature and pH can affect hydrogen bonding and the formation of disulfide bonds. These factors can therefore change the shape of proteins. If the bonds are broken, the shape will change. Hydrogen bonding exists between polar groups. Here we're looking at two different parts of the peptides. I've drawn the two peptide linkages just rotated 90 degrees, so we can see where the carboxylic acid group joined the amino group. If we add on partial charges, we can see that the oxygen and the nitrogen are partially negative, and the carbon and the hydrogen are partially positive. The oxygens will have lone pairs, and therefore there can be a hydrogen bond between the oxygen hydrogens in both cases. The electronegative group means the hydrogens are partially positive. The hydrogen atom is therefore attracted to the lone pair on another one of the electronegative atoms, in this case the oxygen. Disulfide bonding, sometimes called sulfur-sulfur bonding, occurs between the R groups on the amino acid cysteine. Cysteine contains a thiol group. It's essentially the same as an alcohol group, but with the sulfur replacing the oxygen. The thiol group can lose its hydrogen to form a disulfide bond with another thiol group, as shown here, forming a link between two separate groups. These kinds of bonds, the hydrogen bonds and the disulfide bonds, can link different parts of the protein chain together, helping to stabilise its tertiary structure. So for example, we can imagine this represents part of the protein chain, and we might have two, in this case, alcohol groups on two different parts of the molecule. And we can see how the hydrogen bonding between these two parts of the molecule could hold these two parts of the chain together, and how a disulfide bond in this position could hold these two parts of the chain together. We're now going to look at enzymes. Enzymes speed up chemical reactions by acting as biological catalysts. They are proteins, but some also have non-protein components. Enzymes act upon a substrate. Enzymes have an area called an active site, and the substrate fits into the active site so it can interact with the enzyme. The active site is three-dimensional. It is part of the enzyme's tertiary structure. An enzyme will only work with a specific substrate, usually only one. We call this the lock and key model. For an enzyme to work, it has to fit into the active site. The substrate has to fit precisely in. This will form an enzyme-substrate complex. The enzyme will then complete its function, in this case, splitting one molecule into two. This is how enzymes can form new products based on certain starting material. You'll notice the enzyme is unchanged at the end of the reaction, therefore proving it is a catalyst. The active sites of enzymes are stereospecific. This means they will only work on one enantiomer of a substrate. The other enantiomer won't fit properly into the active site, so the enzyme won't work on it. So we say they are stereospecific. Molecules that have a similar shape to the substrate can act as an inhibitor. They compete with the substrate to bind with the active site. No reaction happens when the inhibitor binds. This effectively blocks the active site so no substrate can fit in. So for example, in this diagram, we can see the active site of the enzyme has got an inhibitor fitted straight in, and it matches the shape. This means that the substrate can't fit into the enzyme because the inhibitor is in there instead. And again, this only works because the inhibitor has got a similar shape to the substrate. The amount of inhibition which occurs depends on the relative concentrations of inhibitor and substrate. If there is a lot of inhibitor compared to substrate, it will block the majority of active sites and very little substrate will be catalysed. The amount of inhibition is also affected by how strongly the inhibitor binds to the active site. This is how some drugs work. Some drugs are inhibitors that block the active site and stop it from working. For example, some antibiotics block the active site of an enzyme in bacteria, which makes their cell wall. This causes their cell wall to weaken over time, eventually causing the bacteria to burst. So if you've had common antibiotics, usually penicillin-based ones, this is exactly the method through which they work. They block the active site of an enzyme, causing the cell wall to be not formed properly. The bacteria cell wall can't form properly, it will burst, and the bacteria will die. Since the active site of an enzyme is very specific, it takes a lot of effort to find a drug which will fit into that active site. This is even trickier given that enzymes are stereospecific. It is not just the correct molecule which needs to be found, but the correct enantiomer. The number of enantiomers in a molecule is 2 to the power n, where n is the number of chiral carbons. If you imagine that antibiotics are quite big molecules, you can have quite a lot of carbons in there, therefore a lot of chiral carbons, so you can have sometimes in upwards of the hundreds, maybe sometimes even to the thousands, of possible enantiomers, and only one of those will be the correct shape. Scientists use a trial and error approach. They will test lots of different complexes to see what works best as an inhibitor, and then develop certain ones further. Scientists are now also using computers to model active sites and possible inhibitors. They can quickly test hundreds of molecules to see which will be the best fit for an active site. However, the challenge is then making these molecules, including all of the stereoselectivity, to make sure you produce the correct enantiomer. We're now going to look at DNA and nucleotides. DNA stands for deoxyribose nucleic acid. And it contains all the genetic information for an organism. DNA is a polymer, so it is made up of monomers, and these monomers are called nucleotides. They are made up of 3 parts: a phosphate group, a 2-deoxyribose sugar, and a base. We're now going to look at each of these 3 parts in turn. This is the phosphate group, and this is the 2-deoxyribose sugar. All of these structures are given in the back of your data booklet, so you don't need to learn these structures off by heart. In the back of the data booklet, each of these are printed out for you, should you need to use them in the exam. There are 4 different bases, and again, each of these are printed in the data booklet for you. There is adenine, guanine, cytosine, and thymine. If you've got a data booklet in front of you, or you're making a note of these structured, I would circle the nitrogens drawn in red. These ones are where the nitrogen will bond to the rest of the nucleotide. We're going to come back to that later, but if you're making a note of these now, it might be worth to highlight these hydrogens, as that will help you later on. You don't have to learn the individual structures for the phosphate ion, the sugar, and the base, but you do have to learn how they are put together. So you've got to use the 3 structures given in the data booklet and combine them yourself into the nucleotide, which is the monomer which DNA is made up of. These are the 4 different nucleotides. We've got the adenine nucleotide, the guanine nucleotide, the cytosine nucleotide, and the thymine nucleotide. Each nucleotide is made up of the 3 parts we looked at earlier on. So we've got the phosphate group up here. We've then got the 2-deoxyribose sugar here. And we've got the base here. Now, a moment ago, I told you to circle the nitrogens which were highlighted in red, and they are the nitrogens which attach to carbon 1 on the sugar. So if you know which hydrogens, and you will have to remember which hydrogens they are, but you'll be able to see where they join onto the sugar. That's why we highlighted them in red. So you've got to be able to take the 3 different parts from the data booklet, the phosphate group, the sugar, and the base, and you've got to join them together to draw this structure from the 3 separate structures given in the data booklet. The nucleotides then join together to form a polynucleotide chain. Covalent bonds form between the phosphate group of one nucleotide and the sugar of another. This forms the sugar-phosphate backbone of the DNA chain. So we can see here we've got the phosphate groups in blue, the sugars in yellow, and the bases in green. The backbone is formed through a condensation polymerization process. Here I've drawn out two different nucleotides, and we would lose a molecule of water between the phosphate group on one nucleotide and the OH group from the sugar on a second. So by losing the molecule of water between these two, we'll form a new bond between the O and the P. So we form a new covalent bond between the phosphate group and the sugar. So this will be the structure of two of these nucleotides joined together. We have what is called a phosphodiester bond between the two different nucleotides. We could join on another sugar to the phosphate group on this end, or we could join on another phosphate to the OH group on this end. DNA is formed of two polynucleotide strands, so two of the strands which we've just looked at a moment ago. These are held together by hydrogen bonds between the bases. Each base can only bond with one other base. The bases A and T will bond together with two hydrogen bonds, and the bases C and G will also bond together but with three hydrogen bonds. The way you can remember it is A and T could stand for a table for two, therefore two hydrogen bonds. So if we're looking at a diagram of the structure then, we can see we've got two polynucleotide strands, a sugar phosphate backbone. We've then got our bases in between the two strands, which are held together by hydrogen bonds between the different bases. We're now going to look at the hydrogen bonding between the bases in a bit more detail. You could be given a diagram of two different bases and asked to draw the hydrogen bonding between them. So here we have the adenine base and the thymine base with two hydrogen bonds between them, and the cytosine base and the guanine base with three hydrogen bonds between them. We call this complementary base pairing. Only A will bond with T and C with G, and this is due to the hydrogen bonding between the bases. The atoms which the hydrogen bond is between must have the right polarity and be the right distance apart. The DNA has a twist to it to form that spiral structure, which leaves the bases in the correct orientations and distances apart to form the hydrogen bonding in these specific ways here. You should be able to, from given two different bases, draw the hydrogen bonding between the correct sets of atoms, have lone pairs drawn correctly, and label any partial charges correctly, as well as explaining what I've just explained as complementary base pairing. So explain why only A will bond with T and G and C together. We're now going to look at anti-cancer drugs and how they work. Cancer is caused when cells mutate and start dividing uncontrollably to form tumours. In order for a cell to divide, it has to replicate its DNA. The two strands of the DNA double helix have to unwind so they can be copied. This chemical here is called cisplatin. It stops this happening properly so the tumour cells stop reproducing. The way it works is that a nitrogen atom on a guanine base in DNA forms a coordinate bond with cisplatin's platinum ion, replacing one of the chlorines. A second nitrogen atom from a nearby guanine molecule, either on the same strand of DNA or the opposite strand, can bond to the platinum and replace the second chloride ion too. The presence of this compound bound to the DNA strands causes the strands to kink. This means that the DNA strands can't unwind and be copied properly, so therefore the cell can't replicate properly, and this will stop the tumour growing. Anti-cancer drugs can have side effects though. Cisplatin can bind to DNA in normal cells as well as cancer cells. Any healthy cells that replicate frequently, for example hair cells and blood cells, will be stopped from replicating just the same as the cancer cells. Cisplatin can cause hair loss and suppress the immune system, which is controlled by white blood cells, and it can also cause kidney damage. These side effects can be lessened by giving very low dosages of cisplatin or by targeting the tumour directly. Despite the side effects, it's still used as a chemotherapy drug because the balance of the long-term positive effects, curing cancer, outweigh the negative short-term effects, which will stop once the chemotherapy stops.